When an axolotl loses a leg, its body executes a flawless recovery. It reconstructs every bone, muscle, and nerve, ending with 4 perfect fingers. In a human, the same injury typically results in a permanent patch of scar tissue. Despite these different outcomes, both species share nearly the same deep-seated cellular toolkit hiding inside their DNA. This raises a question about the mechanics of the wound. We need to identify the specific steps the salamander takes at the moment of injury that the human body has bypassed. Regeneration operates as a biological program, a series of dormant instructions that our cells still carry, even if they no longer activate them. The process begins at time 0 with a sudden drop in local oxygen at the wound. This drop triggers an alarm, activating growth-driving proteins like HIF-1 to begin the repair sequence. The nervous system provides the next critical input. Severed nerve fibers must reach into the wound to release proteins that signal the upcoming builder cells to keep dividing. Without this plugged-in connection, the rebuild stalls. This local activity triggers a body-wide adrenaline response. These stress signals prime distant cells and create a systemic memory of the injury, which allows the animal to respond even faster to future attacks. Right at the wound, mature muscle and bone cells de-differentiate, letting go of specialized identities to revert to a younger state. These reset cells multiply rapidly, building a dense lump called the blastema, the engine for the replacement limb. Successfully restarting tissue growth requires this precise coordination between local cellular time travel and body-wide chemical signaling. A blastemal starts as a featureless ball of cells. To form a working limb, these cells must determine exactly which parts to build and where to stop, so they don't produce a random, formless blob. Cells navigate using a retinoic acid map. High concentrations near the body signal proximal parts, fading toward the fingertips for distal structures. A shredder enzyme aggressively breaks down the acid in specific zones, sculpting the precise slope cells use as a ruler. By reading their position on this slope, the blastema cells switch on specific positioning genes, such as the Hox family, to rebuild the limb segment by segment in perfect order. When humans experience a similar injury, the response follows a different path. Instead of forming a blastema, our bodies immediately deploy specialized cells called fibroblasts to the site. These fibroblasts execute a process called fibrosis, pouring out a thick extracellular matrix that cross-links to lock the wound down permanently. This rapid lockdown is a specialized biological strategy, prioritizing the speed of a permanent seal over the slow precision of a rebuild. If our cells still carry the toolkit for perfect regeneration, we have to look at why evolution traded that ability away for thick scar tissue. The blastema represents a significant biological risk. Forcing mature cells to abandon their identity and divide rapidly creates an environment that is nearly identical to the early stages of a tumor. For large, long-lived mammals, the survival math favors safety. Prioritizing fast infection control and strict cancer defense is more advantageous than risking runaway cell growth to replace a limb. Yet, the blueprint remains. Human embryos can heal without scarring, and young children who lose a fingertip can sometimes regrow nail, bone, and soft tissue naturally, provided the wound is not stitched closed. Researchers are now mapping the axolotl's chemical switches to see if we can borrow its logic. The goal is to nudge human healing away from fibrosis and toward the rebuilding process we once possessed. The capacity for perfect regrowth is a latent setting on an evolutionary dial, one that we are finally learning how to read, and perhaps, eventually, adjust.